Why FTD, frontotemporal dementia, is so important for us? It's not a common disease. Probably it's a rare disease, although the epidemiological studies are very rare, few and sparse, with contrasting results. It's important as a neurologist because it involves the higher functions of human brains, what make us humans and not animals. Because when a patient is suffering for this disease, is affected in the social conduct and in the language. Mainly as core features of the disease, first investigators identified the 3 clinical variants: the behavioral variants, the semantic dementia, and the progressive non-fluent aphasia. This, through extensively clinical work and neuroimaging investigation, these variants has been characterized with specific clinical issues and neuroimaging. As frontal variants mainly involved in social misconduct, the loss of inhibition, and apathy or blunting, emotional blunting, and as recognized, mainly being involved in frontal orbital cortex atrophy. The language variants are distinguished in progressive non-fluent aphasia when patients understand the language but are effortful and agrammatic in their speech. And the regions affected in these patients are the frontal left temporal cortices. When patients start with a disease that is deeply impaired in comprehension, they do not understand the name of the significance of simple words, but still presents a fluent language. These patients are labeled as semantic dementia. The different clinical variants have been associated to specific pathological phenotypes. Mainly was recognized FTD as a tau deposit protein, the other half as a ubiquitin positive protein deposition, and later recognized as a TDP43 deposition. It's notably how a part of this of patients affected with language disturbances still recognize a pure adipathology. And this is another open question to understand. But to make the things more complex, great neurologists, including Andrew Kertész, understood that during the disease, the same patient would present different phenotypes, so would impair in motor components of his life. Pyramidal disorder like extrapyramidal disorder like Parkinson's disease, a typical form of Parkinson's disease, or motor neuron disease. I presented this paper that it was a milestone about for FTD neurologist. Here is presented just the evolution of the frontal barrier, but the same it could be for the language disorder. The language through the progression of disease, the patients who present a language deficit presents a different kind of motor impairment. And the different clinical criteria during the time reflected this advancement. And especially in 2001, McCann criteria firstly reported and incorporated the motor variants of FTD, including corticobasal syndrome, PSP, progressive supranuclear palsy, and motor neuron disease. Three diseases affecting mainly the motor dominion of the brain. But it was the genetic that make the differences in this disease as compared to Alzheimer's disease. Why? Because if it's poorly, even if it's poorly understood, it was clear that most of FTD patients has an earlier age of onset as compared to AD, and has a higher positive family history, almost 40%, and up to date, no recognized environmental or risk factors. You know that for Alzheimer's disease, the female gender represents a high risk to develop the disease over the age. Speaking about a genetic risk factor, ApoE4 allele represent a high risk for vascular or Alzheimer's disease pathology. In spite a large search of risk factors for this disease, FTD remains mainly a monogenic disease. The first gene that has been discovered was the same gene that dictated the first pathology discovered. In other words, the tau deposits led to the discovery of a mutation in tau genes. That was in 1897. Through a mechanism by which missense mutation led to a different composition of deposits in brains. Later on, what was called FTLD, ubiquitin positive and tau negative, was linked to a TDP43 deposition as mediated by mutation in progranulin. Another mechanism of genetic defects leading to the same disease, a loss of function, a loss of proteins through an haploinsufficiency mechanism leads to the deposition of TDP33 pathology and eventually causing frontotemporal dementia. The last important genes was the expansion of C9orf open reading frame. Again, another mechanism by which patients may suffer from the same clinical phenotype. And this gene, interestingly, came from the discovery of ALS research, from the research driven from motor neuron disease. What we know now about the neuropathology, we know that tau deposition may be linked to tau mutation, that ubiquitin positive deposition, later discovered as TDP43 deposition, are linked to granulin and C9orf mutation, and more rarely, there is a proportion of rare patients having TDP43 negative, tau negative, and FAS positive deposits. Interestingly, FAS is able, when mutated, to give ALS, but very, very rarely to give frontotemporal dementia. To recapitulate the story about the monogenic, the most important genetic defects associated to FTD, we can say that all affect a population without a gender effect, even if behavioral variants are more prone to be present in male in respect to female. Otherwise, females are probably more prone to present a language disorder, but this result was really not really understood in the few epidemiologic studies that we have in our hands. And the three genes may be distinguished on the basis of phenotypes, being the tau gene more probably linked to extrapyramidal atypical Parkinsonism, the granulin more prone to be associated to language disorder, and the C9orf to behavioral, but also, of course, motor neuron disease, ALS. This just to underline how tau mutations are almost more specific, but not exclusively linked to Parkinsonism. Like otherwise, granulin mutation seems to be more prone to give a language disorder. This is also mirrored by neuroimaging studies, in which the differences that we can catch are characterized by an asymmetric involvement in granulin mutation carriers. Otherwise, for mapped tau mutation, mainly involved bilaterally the frontal cortex. This now is what is accepted as the state of art of these complex disorders. But there are some open questions that we can see in a much more fashionable way in this picture. The open question is that we have here, oh my God. We have here TDP deposition that are driven by C9orf mutation or granulin mutation, but the mutation in TDP43 are able to cause only, apparently, ALS, an ALS disease, apart for a few very rare FTDL cases. The same for FAS. FAS is recognized as deposits in frontal cortices of affected patient by a rare variant of FTDLD, but FAS, when mutated, is able to give a motor neuron disease. As a neurologist, it should be important to understand why tau mutations are linked preferentially to the expression of an extrapyramidal disorders. But we haven't to forget that in small cases, Alzheimer's disease still plays an important role. And these open questions are to be filled, from my point of view, by the molecular neuromolecular biologists, because until now the most players in this field has been the clinician, the neuropathologist, and the genetist, but to identify what should be the fruit of these three, that should be hopefully a disease target for pharmaceutical intervention. I think that it will be the molecular neurobiologist that will be able to provide us a treatment for this disease. Thank you for your attention, and I would like to thank the Associazione Italiana of Malattia Frontotemporale, because it's important to maintain a constant dialogue with the patients and their caregivers, and the people that I'm lucky to work with, that is Professor Padovani and Barbara Borroni and all others that are mentioned here. Thank you again.